Thank you very much. I'm very happy to be here for several reasons, one of which your teachers and I was taught at one time, and so I appreciate the difficulty involved in these things. But also because at the University of Maryland I teach a large undergraduate beginning biology course, and I know what it takes in order to keep students interested. So as was mentioned, I'm a developmental geneticist, so why am I in caves? Well, one of the reasons is that despite more than 100 years of study, the evolution of organisms in caves is very poorly understood. So caves is one of the last frontiers in studying evolutionary principles. And I like to get started here by talking a bit about caves to begin with, and maybe I'll use the technique that was used earlier so effectively today. How many of you have ever visited a limestone cave? Raise your hand. So almost everybody in the room has. How many have visited a wild limestone cave, not on a tour? Somewhat less. How many you visit caves as a hobby and are cavers? So no one, or very few. Well, the point I'm trying to make is that unlike for certain the deep sea habitat and to some extent the high altitude habitats, caves are accessible. People go into caves. Your students may have gone into caves. So caves are an exceptional way in which we can teach students evolution because many students already have an interest in this type of habitat and have visited one and can talk about it. So of course, the obvious difference in the cave habitat from any habitat on the surface at least is the lack of light. And this is a permeating influence. The lack of light causes the absence of a primary producer. Food can be scarce in caves. It can be periodic. Other problems with the lightless habitat is difficulty in finding mates and so forth. So cave organisms would be expected to adapt to all of these changes in order to survive in caves. So what kinds of organisms live there? Well, surprisingly, a wide variety of organisms live there. And perhaps even more surprisingly, from this great group of organisms, they've all converged on a recognizable set of phenotypic features. So we have planarians that live in caves, we have snails, we have insects of various sorts, we have crustaceans, and we have vertebrates, including cavefish, which I'll be talking a little bit more about shortly. And all the things that these organisms have in common, sort of incredibly, if you want to think about it, is first of all the loss of pigment. Cave organisms are albinos, most of them, and are white. And second, incredibly, the loss of eyes. So this has happened multiple times. It must have happened multiple times because these organisms all come from separate lineages, and they've converged upon these phenotypes. Why? And how? So as a developmental biologist, I'm interested mainly in how, and I hope that the answers that I get in asking the question of how will enlighten us, no pun intended, as to why. So Darwin, of course, was one of the first people interested in cave organisms. And he had this is my quote from Darwin, and he had a very interesting thing to say about them, something that is rather surprising, coming from Darwin. So Darwin suggested that the degeneration of eyes in cave animals, and he would have said the same thing for pigment probably, as follows. As it is difficult to imagine that eyes, though useless, could be in any way injurious to cave animals, I attribute their loss wholly to disuse. This is sort of incredible. Darwin is suggesting here a Lamarckian explanation in a book dedicated to natural selection. So Darwin couldn't understand the evolution of cave animals based upon natural selection. And this has persisted to the present day, in fact. We no longer think of disuse as a mechanism for the loss of these features in cave animals, though this is the first one that your students will mention if you ask them why. Cave animals have evolved, probably, because it's the easiest one to think about. We no longer mention disuse because we know that the phenotypes of cave animals are inherited, and there's no mechanism to inherit disuse. So there have been two general theories that evolved themselves at the beginning of the last century to explain the loss of eyes and pigment. The first one is neutral mutation. That is, the genes that are involved in these phenomena under conditions of relaxed selection for light related features in the cave begin to decay, and they continue to decay until their function is lost, and gradually eyes and pigment collapse. So people that believe in this theory think of cave animal evolution as something related to dismantling of the phenotype, continual dismantling from a position in which it started off very similar to its comparable phenotypes on the surface. That would take some time, by the way. And cave animals have not been in caves for very long times in an evolutionary sense, just a few millions of years, because caves actually don't last that long. The other idea is natural selection, but natural selection in a special way, an indirect sort of natural selection generally, in which, for example, things can be selected for that somehow are in conflict with the development of eyes and pigment. And I'll show you some examples of these. So these are the two ideas that have come about. And what I'm going to say today will convince you, I hope, that the first idea is probably wrong, and that the second idea is probably right. But we need a lot more evidence to know the exact mechanisms. So here's the outline of the talk. I'm going to talk first about what is a model cave animal. It's the teleost bony fish Astyanax mexicanus. And I'll give you some reasons for why this organism is the model cave animal. First of all, and then I'll talk about some topics. The first two topics are designed to give evidence against the idea of neutral mutation, that is, complete dismantling of the phenotype in the cave. That's the first two topics, and the third and fourth topics will give you some evidence about indirect selection as the mechanism for loss of eyes and pigment, actually loss of eyes generally, in caves. So that's the outline. So let's go to this model system. So Astyanax mexicanus is a teleost, and the good thing about it is that it has in the same species there are cave dwellers and there are surface dwellers. So the cave dwellers have large eyes for nocturnal feeding. They're pigmented, and the surface dwellers have large eyes and are pigmented. The cave dwellers have lost pigmentation and they've lost eyes. One of the best reasons for working with this animal is that both of these can be brought to the laboratory and studied. It's very difficult for a developmental geneticist to study organisms in the cave, although we try to look at the phenotypes whenever possible in the cave environment. But it's good to have this system where you can bring them back into the lab. And this is one of the few cave animals that can be used as a model system in the laboratory. A second good thing, among many good things, is that we have two forms of the same species here. So we can for a developmental geneticist, this is perfect because you can compare the two as one compares wild type and a mutant. So you have those things. You have other attributes too. Known evolutionary polarity, for example, the cavefish were derived from the surface fish. So we know that what their ancestors like. There's a short time frame involved, about 1 million years. We can use various types of approaches in modern biology, including molecular and genetic approaches. There's a close relationship to the zebrafish, so we can use some things that are already developed in zebrafish in these studies. And as I'll show you next, there is not only one cavefish population, but this type of evolution has happened repeatedly. There are up to 30 different cavefish populations in Mexico, and many of those have evolved independently from a surface fish ancestor. Despite that, I'm just going to talk about one group today, the cavefish from Pachon Cave, which is located about here. This is a map of the eastern Sierra Madre in Mexico. It's the home of these cavefish. Surface fish live in the vicinity as well, and the distribution of surface fish is rather greater, comes up to the southern United States. So, how do cavefish lose their eyes? And I'm going to talk today about eyes, although I could give the same or a similar talk about the loss of pigment. So what happens is cavefish are born with eyes. And it's an interesting developmental phenomenon. It actually looks like a dismantling to some extent, because surface fish and cavefish both have eyes to begin with, or eye primordia, as we say in developmental biology. And in surface fish, the eye continues to grow and differentiate. In teleosts, by the way, eyes grow throughout life as long as they're feeding, unlike us, in which growth of eye stops at the time of gestation. What happens in cavefish, however, is that the eye starts to grow, gets to a stage that's fairly well differentiated in the embryo, and then there's no further development and a gradual degeneration. The eye gets swamped out by growth of the surrounding tissue and is buried deep within the orbit. So if you look at a cavefish and you dissect into the orbit, you'll see a small non-functional eye vestige within a large area of fat within the orbit. So very briefly, how this happens is that at this stage, something happens to the lens that causes it to undergo apoptosis, cell death. We can measure cell death easily by a set of reactions, histological reactions in animals called the tunnel reaction. You can see here, the lens is here, and you can see how it dies. In cavefish, nothing. In surface fish, there's also other places of death. And the lens has a lot of influence on how the rest of the eye grows. So if you kill the eye early, the rest of the eye doesn't grow either. So that's sort of the background as to how the eye disappears. Now I want to go to the first topic. What I want to return to now is the question of complete dismantling of the phenotype. Is this part of the problem under relaxed selection for light? And what I'm going to discuss here is another organ that distinguishes distinguishes light in fish other than the eye, and that's the pineal gland. As you know, the pineal gland has its own opsin and its own secretory apparatus to respond to light. So, in fact, if you look at cavefish and surface fish, and again here we have the ability to look at what is the wild type phenotype in surface fish and the mutant phenotype in cavefish. And we stain animals for embryos for the pineal using a pineal opsin gene. We see that cavefish still have pineal structure. They still have that opsin in the right place, even though the environment is completely dark and the laboratory conditions under which this is being investigated is completely dark. So that suggests that the structure of the pineal doesn't collapse, but does its function continue? And actually, you can do a nice experiment like this. You could do this in a laboratory for students. I've done it. And it's done in the light. So you have larvae of cavefish and surface fish, and you simply pass your hand over the dish where the larvae are. And many teleost larvae have what is called the shadow response. So that shows them a shadow, which is distinguished not by the eyes, but by the pineal. And as a result of the shadow response, here's what they do. I didn't mean to take that turn. They swim up to the top of the chamber as you do, pass your hand over that. I'll show it one more time, if I can. Let's see if I can. So the fish are down, larvae are down here. The hand is passed over the top, and now check to see what happens. So fish swim up to the top. This is called the shadow response. Now, if this happens in cavefish, the pineal is functionally as well as structurally intact. So we do the same experiment with cavefish. They're a little bit slower, but nonetheless, I hope you can see that they move up to the top. So what this suggests is, despite living in the absence of light for more than a million years, cavefish still have the ability to have a shadow response, meaning that their pineal functions. We can go a little bit further with this type of experiment, actually, and we can take out the pineal of cavefish and surface fish and then test this, and we see for the most part we lose these are tracings now, we lose that ability to respond to the shadow response. We take out an eye or two, we have no effect on it. So the conclusion here is that although the selection for pineal function has been relaxed, its light detecting function is retained. In other words, there's no dismantling of that phenotype in the cave. Let me show you another example. The next example is an embryological one. And I've already told you that the lens seems to be a controller for the loss of the eye in cavefish. But what happens if we give it a good lens? We take a lens from a surface fish, transplant it into the cavefish eye, and what happens to its own eye? Is it able to recover and produce an eye, suggesting that things haven't been dismantled, or does it continue to show a regressive phenotype? So we did this experiment, and I'll just show you a movie here. You can see in this movie that a student is transplanting the lens from this case, a surface fish to a cavefish, and we had a zebrafish transplantation in here too. Just to show you the technique. I'll describe, as you're looking at the movie, I'll describe the result. The result is that when we transplant the surface fish lens into the cavefish optic cup, that's the part of the eye primordium that would normally not grow, that optic cup begins to grow. And the lens continues to grow, the optic cup continues to grow. You can see here a cave, and this is only done on one side of the head, so we have the other as a control. When this is done, you can see that the eye on one side is getting bigger. Later on, it gets even bigger. And when these fish grow up, they grow up with a perfect eye on one side of their head. In most cases, this eye can detect a visual stimulus. It's somewhat myopic because the lens and optic cup are not exactly the same size, but nonetheless, it can see. So what this says is that the eye has not been dismantled entirely. All the genes downstream of the lens that are required to make the eye develop are present and potentially functional. And just given a lens stimulus, it can go the rest of the way. So what I hope to have shown by these two examples is that dismantling of the eye or other phenotypes does not occur in the cave, even though they appear to be lost. So what really is occurring? I have to give you one more example about this so I can tell you. And this is another expectation of what would occur in a cave based upon the neutral mutation idea. You expect to see genes being lost and not functioning anymore. But in fact, we see the opposite. When we compare the genes of surface fish and cavefish and their function, we see that genes are upregulated in many cases. Here's a gene on the top. It doesn't matter what it is. It's actually a heat shock factor that's involved in apoptosis, that is the death of the lens. And you can see that as the lens develops during embryogenesis, in cavefish, that gene shown here is upregulated, as shown by that expression zone there. So it's turned on, it's not turned off. There's another case where a gene along the midline, a very important gene that's involved in regulating eye development, called sonic hedgehog, regulates a lot of other things, is upregulated in cavefish so that expression zone spreads out along the midline of the embryo. You can see it larger here. Sonic hedgehog is a very special gene because it not only has negative effects on eye development, so its spreading out would be expected to affect the eye negatively, but it also has positive effects on many other phenotypic features. And that gave us the initial link here, the crossover into going in the direction of what I think explains the entire phenomenon. Some structures are probably selected for in the cave environment for survival, and if they're negatively linked to the eye, that would cause the eye to be destroyed as a byproduct. So that's the next thing I want to talk about. So what are some of those upregulated features? Well, we call them constructive changes and against regressive changes here. And the ones that I'm going to mention now are increased size of the mouth in cavefish and the jaw, and more taste buds. These allow cavefish to better sample their environment, in which most of the food is not all living in the water column now, but is brought in as detritus in cave pools. So cavefish are specialized to search the bottom rather than the water column. And to do this, they've evolved larger jaws, as you can see here, and actually a shovel-shaped jaw. So they go along the bottom like a vacuum cleaner, suck up everything that's there, and then use their taste buds to discriminate what's food and what's not food. The other thing that they've evolved is a set of neuromasts that is a sensory organ, much like the inner ear, but present in much smaller units that line the body of the fish, especially in the head. Cavefish have a large number of neuromasts compared to surface fish, especially in the head. All right, so we're going to take these constructive features and go on with them now. Now I want to ask the question of whether sonic hedgehog is at all involved in the formation of these constructive features. So this is a simple experiment. What we did do is, and you should focus over here, rather complex slide. Surface fish and cavefish, and we look at where sonic hedgehog expression ends up later in development, and it ends up in the mouth and in the taste buds very nicely. So sonic hedgehog is a pleiotropic gene that's expressed both in the precursors of the mouth and jaw and the taste buds, as well as having a negative effect on the eyes. So what happens when you upregulate sonic hedgehog, as happens in cavefish? Well, you get an increase in taste buds and the mouth, and presumably a decrease in the eye. Can we demonstrate that experimentally? So here's an experiment. So the way we do this is we injected excess of messenger RNA for sonic hedgehog in the early fish egg. And this is a surface fish egg, so it develops with an eye, with a lower number of taste buds and a smaller mouth. But as this transformed fish develops, we see some things happen to the eye. We lose the lens. The lens undergoes apoptosis, and we lose the eye. There's an overexpression of sonic hedgehog. And later in development, incredibly, a huge mouth is formed. And that mouth is filled with taste buds. You can see the taste buds are stained individually here. It's quantified here. So the point is, and you can see, there's no leftover of an eye at all in these fish. So what we can do is make a transformation called a phenocopy of cavefish in a surface fish by elevating a single gene, sonic hedgehog, which controls a pleiotropic gene in which eyes are lost at the expense of taste buds. So one of the ways in which eyes have been lost, at least in cavefish, has been to upregulate features that are important for adaptation of the fish to the cave. That is larger jaws, more taste buds, and this is automatically because of the linkages in gene expression throughout early embryogenesis. This automatically causes the loss of the eyes. So that's one way. But it's not the only way this is done. There's another thing that is important, and that is shown by the last topic. There was another constructive phenotype, and that was the increase in number of neuromasts throughout the head. And actually, what these neuromasts do is allow cavefish to be attracted to moving objects. incredibly, and we discovered this by going to the cave itself, you might be able to see some cavefish here. They're surrounding drops from the ceiling because they come and explore those drops as soon as they come off the ceiling. So they have an ability to detect a moving object from a long distance and come and swim to it and investigate. And there's some things missing here, but you can actually stick your finger into the water and get cavefish to follow you if you move it. This is very nice to do collections. I thought it would be hard to collect cavefish, but when I first went to the caves and put the net in, they came to the net and almost jumped right in. Those of you who have experience collecting fish on the surface know that this never happens. You put something in the water, fish scatter, they flee. So cavefish have evolved a behavior here that would be non-adaptive on the surface because of predators. Predators would learn quickly that cavefish would move toward an attraction and eat them. But they can do this in the cave because they're at the top of the food pyramid and there are no predators, no macroscopic predators. Anyway, so how does this behavior function? And here's the ones I wanted to show you, just a few other slides showing how fish come to someone's foot or hand. Cavefish do. You can take this behavior to the lab, and so we simulate it in the darkness. We have fish in a chamber. We can record their activities, and if you do this, record this activity of a vibrating rod sticking into that chamber in surface fish, you see the fish stays away from the rod for the most part. It can't get as far away as it would like to because of the chamber, but it doesn't go anywhere near it usually. Whereas if we do the same experiment with cavefish, the situation is very different. So cavefish come and explore the vibration, actually thinking it's probably food. I want to tell you then that next that this behavior is based upon the presence of extra neuromasts, and we know this for several reasons. One of which is the frequency of vibration that these neuromasts allow the fish to detect is the same as the water drop. And if we inhibit the ability of fish to use their neuromasts, and this can be done with either with cobalt or some drugs, the outcome is that the fish lose the ability to come to the rod. So the final point about this is that these neuromasts develop in the place where the eye would have been. If the eye is there, these extra neuromasts don't develop. So we now come to and they're not controlled by sonic hedgehog, some other gene that we've yet to discover. But there's a space problem here. By getting rid of the eye, it's adaptive to use the space in other means. So this is another way in which the eye has been lost. Indirect selection on an exploratory behavior that is adaptive in the cave because it's good to get to the food fast. And the space is used, which is normally not there because the eye is present, by destroying the eye. So I'm going to give you some overall conclusions here. First of all, cavefish evolution is not the dismantling or degradation of a previously existing phenotype, like Darwin thought. It is the evolution of a new phenotype for adaptation to life in a highly specialized environment. And it occurs by natural selection. The loss of eyes is part of an adaptive program. Second, when cave animals entered caves, they began an evolutionary experiment. And these evolutionary experiments are what I've been telling you. So caves are evolutionary laboratories, and cave animals should provide the materials to study and understand evolution in action, even in students. Finally, adaptations can quickly overcome life in an extreme environment. And we might ask something about this, which I think others have brought up previously. What do we mean by an extreme environment here in the cave? Well, it really has no meaning. It's just a different environment. The cave environment is a very happy environment, probably, for these animals. Importantly, for fish at least, there are no predators. They can evolve behaviors that they wouldn't have been able to evolve on the surface. These behaviors have allowed them to adapt to the cave environment, survive there, and increase and be present in very large numbers, actually. So in that way, it's not an extreme environment to some extent. It's a better environment from the place where they originally lived. So I'll just leave you with that idea and address any questions that you might have.